Alright, this is the second half of the carbohydrate chapter. In the first half we went through sections 1 through 4. Alright, before we begin with disaccharides, I want you to recall what we had talked about with these cyclic hemiacetals. So remember the carbohydrates that we'll be dealing with are hexoses for the most part. So there's 1, 2, 3, 4, 5. There'd be a 6 carbon coming off the top, and then CH2OH. And it's the alcohol group on carbon 5. Right here, the alcohol group on carbon 5. Let me put this CH2OH up here as well. CH2OH. That reacts with the aldehyde on carbon 1. This forms a hemiacetal. So this bond opens up again. We put the alcohol group here on carbon 1. So this is carbon 1. Let me just highlight that. It's carbon 1, carbon 1. And when it cyclizes like this, the alcohol group, right? This is the alcohol group on carbon 5. This is still carbon. I'll put the CH2OH up here. This is still carbon 5 down here. What was the alcohol carbon is now in an ether linkage. It's carbon, oxygen, carbon, and the alcohol is on carbon 1. Right? So we talked about this as the hemiacetal carbon, or the anomeric carbon, the carbon that forms an anomer. This is a new kind of isomer, right? This anomer, new type of isomer. When this cyclizes, the alcohol group can either be down or up. When it's down, we refer to it as alpha. When it's up, it's beta. And the reason this is important is when we start looking at disaccharides and polysaccharides, we will be almost exclusively looking at carbohydrates in this cyclic hemiacetal form. So first off, if we take an aldehyde and we add an alcohol to it, right, this gives us a hemiacetal. That's the name of that functional group. The aldehyde, there we go, the alcohol group. These guys come together. This forms the alcohol. And then the ether. Oops, ran out of room there. So it's carbon, oxygen, carbon. This is the ether linkage. Here's the OH. So the carbon that had the aldehyde, the carbonyl, gets the alcohol group. Whether it's down or up, we call it alpha or beta, and this becomes important when we get into di and polysaccharides. Alright, so for the disaccharides, now, as I mentioned, actually, let's review here for the monosaccharides. The monosaccharides, we had 3. There was glucose, galactose, and fructose. Glucose and galactose were diastereomers. They're both 6 carbons. They're aldohexoses. Fructose is a ketose. We'll see that in just a moment. Those are the monosaccharides. The disaccharides, there's 3 of these as well. There's maltose, lactose, and sucrose. Maltose, we'll see this again later, but maltose is made up of 2 glucose units linked together. So I just abbreviated glucose, GLU, GLU. Lactose is galactose, so galactose and glucose. Remember, disaccharides, we have 2 aldehyde or ketone subunits, so it's 2 monosaccharides joined together. Sucrose is fructose and glucose. Alright, so what we're going to do is join 2 monosaccharides. Remember, we have the cyclic hemiacetal. That's just one of these, say, glucose units. If we add a second, so this is our second molecule of alcohol, you get the acetal linkage. So carbon, oxygen, carbon. put a carbon in here. Carbon, carbon, oxygen, carbon, carbon, oxygen, carbon. It's an ether in both directions. That's an acetal. All right, so an organic chemist would call this an acetal. The biochemist calls it a glycosidic linkage. Biochemists will call it a glycosidic linkage. It's the same thing. A glycosidic bond is the same thing as an acetal bond. The main difference is that a glycosidic bond is what you use to join two separate monosaccharides. All right, again, the disaccharide we'll talk about is maltose. This is formed by two. All right, so remember again, we have the acetal bond. This is our anomeric or acetal carbon. So it's carbon, oxygen, carbon. They don't show it here, but well, they do down here. There would be the sixth carbon is up here. This is carbon 5. A little 5 here. This carbon 6 goes up. CH2OH. But this is the acetal carbon, and notice it's alpha, it's down. This carbon here, this is carbon 4 of the second glucose unit. These are two glucose units linked together. In an acetal, we have an alcohol from carbon 5 combining with the carbonyl, the aldehyde group in carbon 1 forming the hemiacetal. The second molecule of alcohol that I talked about, in this case, comes from carbon 4 of another carbohydrate, another glucose unit. So here's, this is glucose as well, so it's 1, 2, 3, 4. This is carbon 4, this is carbon 4, this is the alcohol in carbon 4. That's what's used to form this linkage here. So here it's down, it's alpha. Here it's beta. So what happens is this glucose forms a hemiacetal and it's beta. The second glucose adds to it, but it adds to it in the beta. Now, one thing that's very important, a hemiacetal, and I kind of did talk about this a little bit, a hemiacetal in the previous one, this is unstable. Remember how the hemiacetal could form and then could open up and reveal the aldehyde, the open chain form, and then it could close down again, and it could form the alpha anomer, and then it could open up, form the open chain acyclic form, and then it could close down again and form the beta form, and those are all in equilibrium. So the acetal is unstable, or sorry, the hemiacetal is unstable. The acetal is stable. So an acetal bond is stable. It does not open up. It is not in equilibrium with the acyclic or open chain form. So maltose does not have a beta 1,4 linkage, beta 1,4 glycosidic bond. It has an alpha 1,4 glycosidic bond, and this is really important. Okay, so this is alpha because it's down, and it's carbon number 1 here of one glucose unit linked to carbon number 4 of the other. So this is an alpha. We refer to this as an alpha 1,4 glycosidic. They call it linkage or bond, glycosidic bond, glycosidic linkage. Okay, that's in maltose, and this is really important because we can hydrolyze. We have enzymes that can hydrolyze or break an alpha 1,4 glycosidic bond, which means we can metabolize, we can digest, and we can metabolize maltose. If it was in a beta 1,4 linkage, we couldn't. So beta 1,4, beta 1,4 is not digestible. Alpha 1,4 is what you find in maltose. Maltose, by the way, I should maybe write this down here as well. Maltose, as the name suggests, is here in Milwaukee County. Maltose comes from malt, which comes from grain, okay, or starches. So starchy food, grains, that kind of thing. That's where you find maltose. Galactose, as the name suggests, you might be familiar with lactose. That's in milk, dairy products. And fructose is found in fruits and sugarcane and honey. Alright, so as I was saying, lactose comes from dairy, milk, cheese, dairy products. Notice galactose here in what? This is a beta 1,4. Beta 1,4 galactose linked in a glycosidic bond to glucose. As already mentioned, maltose, right, comes from grains, and sucrose comes from sugarcane as well as sugar beets, honey, and fruits. And here we have glucose, right? So is glucose linked alpha's down, alpha 1 to this is carbon 2 of sucrose. Carbon 1 is here, 2, 3, 4, 5, 6. Sucrose is a ketose, and so the carbonyl here is on carbon 2, so it is beta in sucrose, alpha's down, right? Beta's up. Beta in sucrose, so it's beta 2, alpha 1, beta 2, and fructose, fructose. As far as the hemiacetals that are here, or the acetals and hemiacetals, remember that this carbon 1 here, this is a hemiacetal. Right? So that's a hemiacetal, and it's unstable. Right? So it can open up, and I ran out of room here. I don't know if I can write. I can't write there. It can open up to give the aldehyde and alcohol back, and because it has this aldehyde, this will reduce the copper 2 in the failing solution. In the process, the aldehyde gets oxidized to an acid. And so something that has a hemiacetal like this, that can open up and reveal the aldehyde, this is referred to then as a reducing sugar. Lactose and maltose are both reducing sugars. They have this hemiacetal. Here, this glycosidic bond here, this is an acetal, right? So carbon, oxygen, carbon. This carbon here is an acetal linkage, carbon, oxygen, carbon, carbon, oxygen, carbon. So there's an acetal here. If we were to go around this guy right there, there's the acetal. Looking down at sucrose, right, here's carbon number 1. This is an acetal, carbon, oxygen, carbon, carbon, oxygen, carbon. So this is an acetal. Remember, acetals are stable. They do not exist in equilibrium, and actually I should write this as an equilibrium. They do not exist in equilibrium with the open chain aldehyde and alcohol, like hemiacetals do. So this acetal is stable. If we look at carbon number 1 here of fructose, it is also in an acetal linkage. Carbon, oxygen, carbon, carbon, oxygen, carbon. So there's 2 acetals here in sucrose, right? So this is also an acetal. Acetals are stable, they don't open up, which means that this is a non-reducing sugar. Right? It will not react positively with Benedict's or Fehling's reagent. It will not reduce copper 2. It will not get oxidized to the acid because it does not. These acetal linkages are stable and they do not open up to reveal the aldehyde and alcohol. Okay, a little bit about artificial sweeteners here. If you look here, sucralose, clearly a derivative of sucrose, trade name Splenda, but it has some chlorines here instead of oxygens, instead of hydroxy groups. All right, so it's artificial. Aspartame, interesting. We'll see this again when we get into the chapter on amino acids. Aspartame is actually a dipeptide. It's a dipeptide. Dipeptide, di means two, peptides means it's made of two amino acids. The first one here is aspartic acid. Don't worry about knowing the names of any of these, but it's a dipeptide, and so that's the first one, aspartic acid. The other peptide in here, and this is phenylalanine. Phenylalanine. And again, don't worry about these names, but recognize it's actually part of a protein. It's not carbohydrate at all. And then there's saccharin, also clearly not a carbohydrate. All right. So we see here some artificial sweeteners, aspartame, saccharin, sucralose, as we just talked about. Much, much sweeter. I believe this is on per mass basis, this relative sweetness, and I'm not sure how they measure sweetness, but it's like on a per gram basis. So sucrose is given as our benchmark. Right, so sucrose is given as the benchmark. And of course, sucrose is composed of what? Glucose and fructose. So glucose is a little bit less sweet than sucrose. Fructose is a little bit more. You put the two together in a dipeptide, sucrose, and you get something that's kind of in the middle. But notice this, if we took sucrose, right, sucrose, which has a value of 1.0, and we hydrolyze it. So there's an enzyme called invertase that hydrolyzes sucrose, and we will get glucose and fructose. Now glucose, of course, is a little bit less sweet, but fructose is a little bit sweeter. Whoops, 1.75. You put the two together, right? You get something that is 2.5 times sweeter than sucrose. And this mixture of glucose and fructose derived from sucrose and hydrolyzed by this enzyme called invertase is called invert sugar. Invert sugar, this is something that candy makers, confectioners use to make their products sweeter by simply hydrolyzing the sucrose and getting this mixture of glucose and fructose. The last one here is this glycoside. I'm not going to try to pronounce that, but the trade name Truvia, right? It comes from the stevia plant. And if you notice here, this is also a carbohydrate. Here we have an acetal bond, carbon, oxygen, carbon, carbon, oxygen, carbon. So this is a glycosidic bond. Here again, it's another glycosidic bond. But instead of being a dipeptide or disaccharide, it is a trisaccharide. So we have 3 aldehyde units linked together. Okay, so now we get to polysaccharides. Poly means many. And for polysaccharides, we're really talking about thousands of these monomers linked together. So each circle here, each one of these rings is a glucose unit. And the 3 polysaccharides that we're going to talk about are all composed of nothing more than glucose. So if you remember, we talked about the monosaccharides. There were 3 types, or 3 different kinds of those. We had glucose, fructose, and galactose. The disaccharides, again, we just talked about those. It was 3 types: maltose, lactose, and sucrose. And here we also have 3 types. We have starch, glycogen, and cellulose. And actually there's maybe another one down here, but for the most part it's just these three. Now starch comes in two flavors, if you wish. There's amylose and amylopectin. We'll talk about the difference of these guys later. Glycogen is sometimes referred to as animal starch. The reason being, it is very much like amylopectin, but it comes from animals. Starch, these guys come from plants. Right? So grains, cereals, and whatnot is where we get starch from. And remember, let's go down here. So if this is a starch molecule, if we have a dipeptide here, they're actually showing this. Yeah, this is, I was going to say, this is cellulose down here. You can see this is cellulose. This is a beta 1,4 linkage. But if it was, if these were all alpha, this would be maltose, right? Maltose is just a dimer of two glucose units linked alpha 1,4. So if it was alpha 1,4 like that, it would be maltose. So maltose, of course, is derived from starch. Maltose also comes from grains. Cellulose here, this is plant fiber. So it's what the plants use as a structural component. And here we have the cell wall, which is composed of bundles of this cellulose, and so it's forms the leaves and stems and the trunks of trees and twigs and whatnot. And we cannot eat this, right? We do not. This is not food for us. Now, there's some animals that do eat this kind of thing, ruminants such as horses and cows and deer and whatnot, but not food for humans. So I'll put humans here. We'll talk more about that in a little bit. Okay, but cellulose is linked. You see here, alpha 1,4. I'm sorry, beta 1,4. Beta 1,4. Maltose is alpha 1,4. Amylose and amylopectin, glycogen as well. These are all linked. These first two, starch and glycogen, they're glucose to glucose, and again, alpha 1,4. All right, again here we see cellulose in this beta 1,4 linkage. This is carbon 1, 2, 3, 4 of the first unit. This is carbon 1 of the second, so it's alpha 1,4. I'm sorry, it's beta 1,4. Alpha, this glycosidic linkage, this is what you see in both kinds of starch and glycogen. Okay. Now, we have, like I mentioned before, we have an enzyme that can break this bond, so this can be hydrolyzed or broken, which means it can be digested, right? So it's digestible. We can use this for food. We do not have the enzyme that hydrolyzes beta-glycosidic bonds. And in fact, even cows and horses and whatnot lack this enzyme that breaks this, but they have a much more complicated digestive system, and they have bacteria in their gut that actually hydrolyze this bond. So they're able to get some energy from cellulose, from eating leaves and grasses and whatnot, whereas we can't. We have to only rely upon starches for that. So here's amylose. We refer to this as a linear form of starch. And then there's amylopectin. Remember, starch had two flavors. I call them flavors, or varieties if you wish. Amylose and amylopectin. And the thing about amylopectin is it contains these alpha, this is down again, this is alpha 1, this is carbon 6, and it contains alpha 1,6 branches. So amylopectin is branched, right? It's the branched form of starch. Amylose is not. It's not branched. Not that this makes a big difference for us in terms of how we get energy from them when we eat starches or grains. but it does make a difference in the lab, and that has to do with the iodine starch test, or the reaction between iodine and starch, and we'll talk a little bit about this in a moment. All right, so here we see amylose, and again, this is unbranched. Amylose is unbranched, and it forms this regular like spiral staircase or helix. So it's helical and not branched. Amylopectin, however, is branched, and it's branched about every 20 to 25 glucose units. Glucose monomers or glucose subunits or whatever. Okay, so if you were to count here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, you get to about 20, there's a branch, you get another maybe 20 to 25, there's another branch. And because of this, the helical sections in amylopectin are shorter than that of amylose. Amylose is kind of like what we saw with cellulose. It's completely helical, unbranched, or linear, right? So it has longer helices, and because of the branches here, it has shorter helices. The amylopectin has shorter. Right? Now, where this comes in to be important in the laboratory, iodine starch test. So put it down here. We have starch, which would be maybe a cloudy solution, because starch, being a polysaccharide, is not very soluble. Something I forgot to mention. Because these are so long and they form these molecules are so large, they really aren't soluble, which is a good thing, because you don't want, if it rains, you don't want the leaves and grasses to dissolve, right? So polysaccharides like starches and cellulose are insoluble. But we take starch and in solution, and it's cloudy again because it's not very soluble. And combine that with iodine. Iodine is diatomic, right? And it's red, right? So it has a red color. You put these together, and it turns into a blue or purple complex. So what happens is iodine is red, but when it binds to starch here, it kind of fits inside this helix, and it forms these polyiodide chains. Polyiodide. And when it does that, it changes colors from red to blue or purple. Because the longer helices in amylose, it forms a different color than that of amylopectin, because amylopectin has these shorter helices. So you can actually tell the difference between amylose and amylopectin based upon the color of this purple starch iodine complex. We get to glycogen. Again, glycogen is referred to as animal starch, and the reason for that is very similar to amylopectin. It has these alpha-1,6 branches, and I'll just say here it's branched. But the difference between amylopectin and glycogen isn't just because glycogen comes from animals, amylopectin comes from starch. Animal starch, or glycogen, is branched about twice as much, so branched about every 10 to 12 glucose units. And what this means is it's what we call highly mobilizable. And what do we mean by highly mobilizable? Again, we have enzymes. Now, first off, glycogen is a storage form of glucose for animals. Storage form of glucose. Okay, so you might be familiar that with the idea that one must maintain a proper blood glucose level, and if you don't have if you don't have enough glucose, you become hypoglycemic. So there's hypoglycemic, and really bad things can happen if you become too hypoglycemic. You get irritable. People talk about being hangry because they're hungry and their blood sugar drops. It affects the brain. The brain, by the way, brain and active skeletal muscle. I should write that down. Brain and active skeletal muscle rely on glucose primarily for their energy, for their food. So if your blood glucose drops and you become hypoglycemic, your brain is affected. Now, the active skeletal muscle and brain, they can use other carbon sources, but by far the preferred source is glucose. So this can lead to, you can become dizzy. Your hands can get sweaty. Your thinking is impaired. Eventually it can lead to a coma and death if blood glucose gets too low. So it's really important to maintain proper blood glucose, and that's what glycogen is there for. So glycogen is stored primarily in liver and muscle, and when I say it is highly mobilizable, what that means is in order to use this one glucose molecule, we have to cut it off from this polypeptide. Okay, so if you imagine if we only had, let me put a big long helix here. These glucose molecules are just lopped off one at a time. They're just lopped off one at a time, starting at one end. Well, if it's branched, that means we've got 1, 2, 3, 4 different ends maybe, and it can be hydrolyzed faster, and we can get the glucose into the bloodstream faster, so our brain and skeletal muscle can use it. Plants, they have amylopectin, but they have no need to they're not going anywhere, so they have no need to have a highly mobilizable form of glucose, whereas animals do. One more thing about, I'm going to put a helix down here. So if you have a polysaccharide here made up of dozens or hundreds of glucose subunits, on one end, remember these are glucose to glucose linked alpha 1,4. So on one end you're going to have a glucose molecule like this, CH2OH, and okay, you've got the alcohol groups here. On the other end you have a glucose subunit. Okay, this bond here is an acetal. Right? We link one glucose to another in an acetal. This one here is a hemiacetal. Remember, hemiacetals are unstable and they can open up, and when they do, they can react with copper in Fehling's reagent, and so this end here is referred to as the reducing end. Right? Because this is the end that contains the hemiacetal. This end here is the non-reducing end. So these polysaccharides, they do have a reducing end, but remember there might be hundreds or thousands of these glucose units and only one hemiacetal. So because of that, it's really a concentration effect. Polysaccharides are considered non-reducing even though they have a reducing end, because there's only one molecule here that can open up and get oxidized by Benedict's or Fehling's reagent, and score positive in that test. All these others are linked by acetals. So there's many glucose units linked through acetals, and only one has the hemiacetal. Okay, there are other places where polysaccharides are used. So other uses of polysaccharides. Okay, there's hyaluronate or hyaluronic acid in the vitreous of the eye. These two are actually, I don't know if they have the molecules mixed up or they just have the names mixed up, but I'm going to, chondroitin, you might be familiar with. Sometimes people will take that as a supplement for their joints. And heparin is a anticoagulant found in the liver. Anticoagulant. They are polysaccharides, but they've been modified, right? So they have sulfates, or this is an amide. So they form a protective role, chondroitin, right? Protective role. Hyaluronate, I'm not sure what role you call it. I guess it would be a structural role. Structural role in the eye. There's also a protective role. I don't have an example to show you, but a protective role for carbohydrates would be the what we call glycosaminoglycans, and these are found in mucus, or so actually another name for these are mucopolysaccharides, and they are found in the nose and in your airway, and they also form a protective role. So there's many uses for carbohydrates: a protective role, a structural role. Here in your joints, I guess that would be kind of a, I don't know if you want to call that structural or protective, kind of both. It protects the joints. If you happen to be an arthropod, so like a beetle or an ant, beetles especially, I don't know, grasshopper, something like that, they do not have skeletons made of bone. They have exoskeletons. Right? These exoskeletons are also made up of a polysaccharide called chitin. So clearly a structural role for these insects. We have a recognition role. All right, a recognition role for carbohydrates. You're familiar with the blood groups. Blood groups type A, B. There's also AB, type A, type B, type AB, which has both of A and B, and type O, which has neither. And the difference between these are on the cells, and you'll see this later on when we get into the chapter on lipids. So this is, say, a blood cell. There are polysaccharides that kind of decorate the surface of the cell, and depending upon which polysaccharides are present, that's what blood group they belong to. So the organism can recognize self versus non-self based upon what carbohydrates are decorating their cells. You don't have to know the particular monosaccharides that go together to form these, or the order or anything like that. All you need to know is that the blood groups are differentiated based upon what carbohydrates are present, and that's another role for carbohydrates in this recognition role. Okay, so that does it for the chapter on carbohydrates. Next up, we'll be talking about lipids.